What are the essential factors for photosynthesis? Already we know that carbon dioxide, water and chlorophyll. So here by the process of photosynthesis, the plants are converting solar energy into chemical energy. Solar energy into chemical energy. So here the solar energy, nothing but sunlight. These sunlight are converting into chemical energy by the process of photosynthesis. Then what are the factors, essential factors? Here first one is the water, carbon dioxide and chlorophyll. Next one, water and photosynthesis. So Van Helmont is one of the famous scientists. He conducted so many experiments. He conducted one experiment related with water is essential for photosynthesis. He conducted experiment with a small plant. He taken a small plant and planting in a small pot. Before planting, he measured mass of dry soil which added to the plant. Then same thing, mass of plant. Then he left this setup or this plant for 5 years. He given only rainwater to the plant. After 5 years, he measured the mass of dry soil and mass of plant. So after this experiment, after 5 years, he confirmed that plants needed water also, not only soil. Plant need water for their growth. He concluded that water is essential for plant growth. Next one, air and photosynthesis. Joseph Presley is one of the famous scientists. He conducted series of experiments to prove the air is essential for growth of green plants. He conducted experiment in the year 1770. He proved that air is essential for growth of plants. So in these experiments, he discovered the air which is useful for survival of animals. It is nothing but oxygen. In the year 1774. Later, in the 1775, Lavoisier given the name oxygen. So the Presley performed different types of experiments. Taken a jar and taken a burning candle and mouse inside the bell jar. After a few minutes, the burning candle extinguished and the mouse suffocated and died. So from this experiment, he concluded some gases are released by mouse and burning candle, it will damage the air. Then same thing, the continuation of this experiment, that is, he kept mouse and burning candle and mint plant inside the closed bell jar. In this experiment, after few minutes, mouse is alive, candle continually burning. From this experiment, he concluded that plants are absorbing the damaged air which are released by the mouse and burning candle, and these plants using gas which are essential for the growth of mouse and the burning of candle. So these essential gas which are helpful for the survival of living organism is nothing but oxygen. So he concluded that is air is very essential for the growth of green plants. Exchange of gases. Exchange of gases between plants and animals and burning of candle in this experiment. So here the exchange of good and bad gases. Here the exchange of carbon dioxide and oxygen inside the bell jar between plant and the burning of candle as well as the moss. So these exchange are taken place in the leaves. Why? Because in the leaf we have some pores like structures. These pores like structures are called as stomata. These stomata are consist with chloroplast. Next, activity number 2, CO2 is necessary for photosynthesis. It's also called as moles half-leaf experiment. We need transparent bottle, potted plant, cavite solution, splitted cork, iodine solution. Next procedure, take a potted plant and keep it in the dark nearly a week for the removal of starch. In this time, no photosynthesis taken place in the plant. So, the stored starch utilized by the plant, and in the plant there is no starch. After this, take a wide mouthed transparent bottle and pour cavite solution into the bottle. So, this cavite solution absorbs carbon dioxide which is present inside the bottle, and insert one of the leaves through a split cork into the bottle containing KOH solution and applying grease or Vaseline on the bottle to keep airtight. So leave the plant in sunlight for 3 to 4 hours. Then after 3 to 4 hours, test this leaf and other leaf of this plant for presence of starch. Observation. The part of the leaf outside the bottle turns blue-black because starch is formed. But inside the bottle does not turn blue-black because the carbon dioxide is not present inside the bottle. Why? Because a KOH solution absorbs carbon dioxide which is present inside the bottle. So here the outside the bottle carbon dioxide is present, so the starch is formed. Result of this activity, it proves that carbon dioxide is necessary for photosynthesis. Next precautions. The part of the leaf kept inside the bottle should not touch KOH solution. The operator should be kept airtight by applying grease or Vaseline. Then for destarching the plant, it's kept in the dark for nearly a week to remove the starch from the leaves. Then only we can understand that the starch is prepared in the leaves or not. Next, light and photosynthesis. John Ingenhousz is one of the famous scientists. He found the way in which plants form oxygen. In the year 1779, he noticed the oxygen forms from the plants in the presence of light only. So he done an experiment to prove this. He done experiment with an aquatic plant like hydrilla. He observed that in bright sunlight, a small bubbles formed around the green parts, while in the dark they did not form. So he confirmed that the bubbles are formed in the presence of sunlight only. From this experiment, he also found that the gas present in the bubble was oxygen. It was further confirmed when Engelmann in the early 20th ingeniously detected the point of maximum rate of photosynthesis, and also more studies on effect of light on photosynthesis, the role of different color compounds called pigments in plants, and the utilization of light energy. Next activity, oxygen is produced during photosynthesis in the presence of light. We need hydrilla plant, beaker, funnel and test tube. Procedure of this activity, just arrange the apparatus as shown in the figure. Make two identical sets. Then place some water plant hydrilla in a short stemmed funnel and keep it in a beaker. Just to see this figure. Then pour the water in the beaker. Then invert a test tube full of water over the stem of the funnel. You can observe here. Ensure that the level of water in the beaker is above the level of stem of the inverted funnel. Then place one apparatus in the sun and other one in the dark for at least 2 to 3 hours. you would see that in a place of water there is air that fills in the setup kept in sun. It is actually a gas that will be collected in the test tube. They observe the other setup kept in dark. Is there any difference in the amount of gas collected? Yes. There is no bubbles in the dark plant. So test the gas in the test tube by inserting a glowing matchstick or incense stick which would burst into flames. This shows the presence of oxygen. Precautions. Setup should be kept in sunlight. The level of water in the beaker should be over the level of stem of the inverted funnel. Then while removing test tube, place your thumb near mouth of the test tube to prevent escape of gas that is produced in test tube, nothing but oxygen.